Bacterial wilting is the wilt disease in plants that is caused by some xylem colonizing pathogenic bacteria. Solanacearum is a pathogenic bacterium that causes severe wilt in many plants. The symptom of bacterial wilt appear on foliage of plants. The symptom of wilt disease is wilting of leaves at the end of plant branches. It is a gram-negative, rod-shaped anaerobic bacterium. It is a soil-borne bacterium. It can be culture in liquid and solid, agar, medium. It causes bacterial wilt disease in over 200 families of plants, including crops as potatoes, tomatoes, peanuts, tobacco, bananas, and plantains, as well as many native plant species. It primarily enters the plants through the roots, colonizes the xylem tissues, clogging the vessels and eventually leading to wilting and plant death. It is destructive. This bacterium kills any host plant it infects. It invades and colonizes the host xylem tissues, leading to the obstruction of water transport, plant wilting and eventual death. And it has a wide host range which includes some very important crop plants. Areas affected by bacterial wilt incur/suffer significant economic losses due to reduction in agricultural produce. Moreover, the pathogen is persistent and difficult to eradicate once established in the field. Co-occurrence pattern of average rainfall and Ralstonia wilt incidence during 2004 to 2005 in West Bengal showed the relationship between monthly precipitation and Ralstonia-Solanacearum wilt incidence in tomatoes. During periods of low rainfall or low soil moisture conditions, there is reduced Ralstonia wilt incidence when compared with high rainfall or high soil moisture conditions. The pathogen species is distributed throughout the globe. The species has an extensive genetic diversity, and strains from different regions are genetically distinct from each other. Because of its distribution and complexity in genetic makeup, the pathogen is now popularly known to belong to a complex called the R-Solanacearum species complex, RSSC. This diversity makes the pathogen virulent to diverse host plant species. What strategies are currently used to curb the disease? Cultivation practices. Biological, physical, chemical, cultural practices as control agents. Avirulent strains of Ralstonia-Solanacearum, Pseudomonas species, Bacillus species, and Streptomyces species are well-known biological control agents. Resistant genotypes. Transgenic systems. Focus of research on bacterial wilt disease in NIPGR. Dissecting the molecular and physiological responses of plant to Ralstonia-Solanacearum and an accompanying stressor. Screening whole genome-wide transcriptomic data for genes involved in resistance response to the pathogen. Understanding plants' reactions to individual Ralstonia-Solanacearum and drought stresses, and to combination of these stresses.